Is it? Okay. So hi everybody. Week 7. This is, we're done with unit 1. We're moving into unit 2. This is Bio 208 with Dr. Granier. The crew is here for lab, and today we're going to do a quick review in lab based on our lymphatic cells, tissues, and organs. All right. And so remember that our cells are going to be, where do we have the lymphocytes? Okay. And so remember that from our blood board, all right, we learned that the lymphocytes are going to be represented by these 3 cells. Okay. And you can't tell the difference by the naked eye what those 3 cells are. Okay. And we learned that the lymphocytes come from the lymphatic progenitor cell. And that's usually because we don't have EPA. We don't have colony stimulating factors. And what happens is that lymphatic progenitor cell can give rise to our T cells, our B cells, and our natural killer cells. And we went through this in the lecture that our T cells can become potentially cytotoxic T cells. They can become T helper cells, T suppressor cells, or T memory cells. Okay, and our B cells, eventually they start off as these naive, they're not sensitized yet. And after they are sensitized, they can become plasma or clone cells, and they're amping up and ramping up the antibody distribution that then is spreading out. And then a few of them are going to become memory cells. All right. And these cells tend to be in our lymphatic tissues, our lymphatic organs. Okay. And they each have a role in, specifically like our T helper cell, they are the bridge between the non-specific defenses and the specific defenses. Okay. And just to review again, because our white blood cells are part of the immune system, remember this big cell with the kidney bean shaped nucleus? That is your monocyte or your macrophage. That cell is one of your antigen presenting cells. Its job is to phagocytose, so it can then digest, pull some of the pieces and parts out, present it to our T helper cells, and our T helper cells can determine if what that antigen presentation is, self or not self. All right, and then they can potentially ramp up B cells and T cells to a new antigen if the macrophage monocyte presents a new antigen. Okay. Our next one is labeled number 4, and I'm trying to point to all of them. This is, like it says, the most numerous. This is your neutrophil, polymorphonuclear, granulocytes. All right. And your neutrophil, and then this one with the red is your eosinophil. Those 2 are classified as macrophages, and they are supposedly spitting out their contents to break up a pathogen, to break up some cell debris. They ingest pieces and parts of that cell debris. They can then potentially present to a T helper cell and be then triggering a response from the specific defenses if they show that they found a new pathogen and antigen presentation. Okay. On here, we can't tell the difference between our lymphocytes. All right. So what we're going to learn later on is, in order to tell the difference between our lymphocytes, you have to do specific tests to the membrane proteins. And you have to find which of these cell membranes are expressing a protein that we label CD4, and which of these cells are expressing a protein known as CD8. And once we know what those proteins are, we can start to differentiate. This is a type of T cell that does this. This is a type of B cell that does this. Okay? And we'll get to that in specific defenses. So our lecture probably on Monday. But for lab purposes, this will be on your lab exam again. If you didn't learn it the last time, you have another chance. So everybody needs to get back into learning what are the cells? And just like last time, we're not going to be able to differentiate between our T cells and our B cells and our NK cells. So you can just tell me it's a lymphocyte. Okay? And again, when you get into our lecture from Monday, remember that you are expected to tell me what are the role of cytotoxic T cells. We expect them to look for cells displaying an antigen. The antigen that it's looking for, it has been tagged and it's been told, if you find a cell with this antigen, kill it. That might mean the entire cell is a foreign particle, a foreign pathogen. That could mean it's a cell that's self, and the particle that is displayed on its membrane, the antigen that's displaying, is because a virus is inside or some type of parasitic host is inside and helping to create inside that cell these antigens that are now showing up on their membrane. Okay? So cytotoxic T cells kill those cells. Suppressor cells try to prevent them from going overboard. Memory T cells are going to retain, okay, here is this antigen, maybe like I was saying on our example, this is the Cubs logo. So anytime I now see a Cubs logo expressing cell, I know to kill it, because either it's a foreign entity and it shouldn't be here, or it's one of my self cells that have been invaded by a pathogen, invaded by a virus, and are now making Cubs logos, and they shouldn't. Okay? All right. Remember, the B cell, when it's naive, it still has all in its genetic code, all of the ability to make immunoglobulin A, immunoglobulin G, immunoglobulin E. All right. And most of those immunoglobulins, you would do big I, little g, and then the capital letter. Immunoglobulin A, immunoglobulin E. Okay? And that means in its DNA, somewhere in there, there's like the A intron, the G intron, the M intron, the E. And the naive B cell still has on its 5' to 3' DNA strand, it still has those areas to make all of these different type of antibodies. Right? And once it becomes exposed to the pathogen, and again, that's where that T helper cell probably plays a role here, somehow magic happens, and it's going to go, I don't need you, I don't need you, I am going to keep these two, and I'm going to make an IgG or an IgM, and that's going to be the antibody that I make with special pieces in here that I'm going to tweak that will be able to find my pathogen because it's expressing that Cubs logo. And it'll always be able to find the Cubs logo. I don't know how it works, but that's microbiology, immunology, and cell biology, and that's above what we want to worry about. Okay? But once I make that cell that now has that, what is it? I said G and that M, right? It is forever going to make antibodies that always have those pieces to it, and only those pieces, and it's always going to be looking for my Cubs logos. Okay? And remember, one of them every few often times will remain and continue to thrive, so the next time we have the infection, they can ramp up against it. Okay? These antibodies, they do not, I don't know how to, they do not kill. All they do is attach. So if a cell is attached with antibody, either my cytotoxic cell can come in and kill it, my macrophage, my microphages can come in and kill it, because now we know antibodies are attacking it and binding it, that it's a bad cell. It's a cell that's gone rogue, gone hijacked, or doesn't belong. It's just the antibody can't kill it. But the antibody could maybe find that little hiding spot, because the antibody's small enough that it can find all the little hiding spots and bind to things and make something have like a big flag on it. Okay? All right, remember these guys are part of your non-specific system, and they are all about surveillance. And they do not go to the thymus gland, but they are immunocompetent. So they are our evidence that you can get immunocompetence in bone marrow, and you don't have to go to the thymus gland. Okay? And they're going to work a little bit differently from these guys on how they kill things and how they destroy things, because they are going in, like I said, they're like the door-to-door salesman. They're going to go into a neighborhood. They're going to go into an organ. They're going to go into an area, and they are going to knock on every door, every cell. And the first check, self, not self. Second check, normal function, not normal function. Third check, normal function cell, but hijacked by some weird thing that it's producing because it's no longer all of our DNA in there. And it'll kill it. Okay? All right, so let's move to our tissues. Okay? On our tissues, I'm going to give you the picture. And in your lab manual, it's a little girl picture, and it has the 3 pairs. You have to adequately label those 3 pairs. All right? And it says, I think, the pharyngeal tonsils. You can give me the adenoids for that. I'll accept that. And then you have the, what is it? The lingual tonsils. Is anybody on that page?
Yeah, lingual.
And what's the third one?
Pharyngeal.
Pharyngeal.
Yeah.
What's the other one?
It's right and left palatine tonsils.
Okay, so the pharyngeal and the adenoids are the same. Palatal, because it's more around the palate, and then the lingual. I think that's the right thing, okay? Now, don't get those confused with your salivary glands, because you have 3 pairs of salivary glands. And those are the parotid, sublingual, and submandible. And again, they're going to be in and around the tongue, the lower areas, because they're trying to push fluid into the mouth. Okay? All right. So expect a station on your lab exam where you have to, on the picture of the little girl, adequately identify the 3 pairs of the tonsils. I might only ask you 2, but you got to know all 3, because you don't know which 2 I'm going to ask. Malts and Peyer's patches. Again, in your, we don't have any models of this, so this is more lecture, but in your lecture, again, you're going to see there's embedded in the digestive histology submucosa areas, there's going to be this big population of lymphocytes that kind of come together. And then there'll be these little kind of pathways to other areas of lymphocyte-rich and macrophage and monocyte-rich neutrophils, eosinophils, and basophils-rich areas. And they work to, if something gets, again, kind of past and tries to get into our system through the large intestine. So they try to get in with food or through the back way, all right? That's why little kids stick their fingers up their butt, you know. We don't like to stick things the back door up our butt, but that's an entryway for things to potentially get into us. That these are there to try to, again, if it get past the first barrier, they're there to try to capture it and remove it. And then if they get past these, they still have to get through the lymphatic vessels and the lymphatic nodules or lymph nodes before they still make their way back into the cardiovascular circulation. Okay, our organs of the lymphatic system, we have the spleen, all right? And again, how is an organ different from a tissue? It's got a capsule, some type of connective tissue capsule. It has a blood vessel entry and exit point, so therefore also lymphatic and nerves enter and exit, all right? And inside, it's going to have a high population of my lymphatic cells, along with other white blood cells. And in the spleen's case, blood is going to flow through this. So how is this guy different from all my other lymphatic vessels and tissues and organs? No lymphatic fluid, blood. So my lymphocytes, my macrophages, my white blood cells, they are getting to filter blood in the spleen. And this is one of the places we directly get to take pathogens out of blood. So that's what makes it unique. The thymus gland, remember, we don't have a model for it, but it's going to be a lymphatic organ because it has a capsule. It's going to be full of some epithelial glandular cells that are going to create a little pod. All right, so the cells are going to create like a little area that inside I can put developing T cells, and I can secrete what's known as thymolytic hormones to influence these T cells to mature and become immunocompetent. And then when they get competent, they can go back out, jump in the blood, and then travel to their desired final location. Okay, so the thymus gland is not dealing with pathogens immediately, but it's where most of your T cells are going to have to go and develop, become immunocompetent, leave, and then go to their organs, go to their location, their tissue that they're going to take up permanent residence to fight as part of the immune system. Okay, so it's a little bit of a unique organ in that it's not directly filtering lymphatic fluid. It's a place where we mature T cells, okay? And our last organ we have to talk about are our lymph nodes. And on our model, I'm gonna try to turn the camera and bring him over, okay? They're usually, again, shown as these green nodules and the tubes. So remember that the vessels are going to be named, if they come into a lymph node, for this specific lymph node, they are going to be known as afferent vessels. And then the 1 to 2 vessels that leave will be an efferent vessel for this lymph node. And very quickly, an efferent vessel can turn into a afferent vessel onto the next lymph node. And eventually these lymph nodes are going to gather and gather and gather. There'll be that cisterna here that helps kind of suction it up. And then that'll work with the lungs and breathing to help try to get, again, that lymphatic fluid through the thoracic area. And we're trying to get to these 2 locations, and I'm trying to make sure you guys can see. All right, remember where the jugular and the subclavian vein come together? I don't know if it's, see that green? Y'all can see that over there too. All right, this little doorway is known as your thoracic duct. The one over here on the right side is known as your right lymphatic duct. And the fluid that we've spent all of this time and energy to go through lymph node after lymph node after lymph node after vessel after lymph node after vessel after lymph node, have to remove the cells, remove the molecules, and then reduce and remove microparticles, removing out bacteria cells, have been nature, it's part of the system, it's right here, it's right the lung, it's just the left part of the rest of them. Okay? And currently lymph nodes is every good, because there's an outnode here, the line is moving right in, and the infected console is up. And that's why because the cells are going through mitosis, and they're going through cells, and most of the cells, maybe cells, we make a lot of T's, that's what we do. Maybe a lot of people appreciate, hey, these cells react, maybe they more or less. And how these cells, exactly like this, there's a lot of cycle cells, there's a lot of growth in cell number, and a lot of growth in fluid coming in, cytokine theory, so osmosis numbers, not hypostatic, that's just as well. Right? So here's what you have. So how do we expect your lymph node and or the station on your left hand? Also for sure, a